Hi everyone, this is the IGCSE Human Biology 2023 May to June paper 1, and I'm just continuing on from question 5. If you want to look at part 1, you can look at the link in the description below. Okay, let's get started. Okay, question 5. Pit latrines can be used to get rid of urine and feces. They can be used where the soil is permeable to water and are always placed downhill from wells. Part A1, explain why a pit latrine should be placed downhill from wells. So if you can imagine, if you've got a hill, this would be your well, and this would be your pit latrines. And it's important for it to be downhill, because if it was the other way around, like if it was this way around, so pit latrines and this is the well, what would happen is you would have the urine and the feces would come here, and because I think they mentioned here that the soil is permeable to water, if it was raining, you would have all that rainwater mix in with the feces and the urine, and that rainwater will carry the feces and the urine to the well. All right, so let's write that down. It is placed downhill so that when it rains, the water does not mix with the urine and feces and carry it downhill to the well. Alright, part 2. Give the reason why pit latrines must be placed in soil that is permeable to water. The reason why is you want to allow for that waste to be absorbed by the soil. This is because the waste can be absorbed by the, so we can say liquid waste as well. All right, let's move on to part 3. Explain why, when a pit latrine is not in use, the latrine should have a cover, and why that cover should have a long handle. So we can split this answer into 2. We can talk about why there should be a cover, and the obvious reason is to prevent the smell from coming out. Another thing is to prevent organisms like flies or things that are attracted to decaying matter from going into the pit latrine. And that's important because flies, for example, can bring along with it bacteria or other pathogens that can cause diseases. Now, the second part is why they should have a long handle. And that is because if you have a long handle, your hands are going to be away from the entrance of that pit latrine, which means you're less likely to come into contact with harmful bacteria because there's going to be a lot more bacteria at the entrance of that pit latrine. So we can say it is important that it has a cover so that the smell does not come up. It also prevents organisms like flies from entering the pit latrines. Also prevents animals are attracted to waste, such as flies, from entering. These flies can carry pathogens and therefore spread diseases. Right. The reason why the cover should have a long handle is to keep a person's hands away from the entrance of the pit, as the pit would likely contain harmful bacteria. All right, let's go over the marks. The first mark is to prevent smell. The second mark is prevent entry of flies or rats. So I wrote that there. That's the second mark. The third mark is they can carry pathogens. And the fourth mark is spread diseases. There's a fifth mark to keep hands away from the entrance of the pit. And the last mark is because the entrance of the pit would have more pathogens or bacteria in that area. All right, let's move on to part B. The concrete slab shown in the diagram is washed regularly with an antibacterial solution. A scientist wants to investigate two antibacterial solutions to see which solution is more effective. Design an investigation the scientist could use to find out which of the two antibacterial solutions is more effective. Include experimental details in your answer and write in full sentences. So whenever they ask us to design an experiment, we have to consider the following things. Number one is the independent variable, which is what is the thing that we're changing. In this case, we are trying to investigate two antibacterial solutions, right? Then we want to look at our dependent variable, which is what are we measuring and how are we going to measure that? Then we're going to look at our control variable, then talk a little bit about the method. And then lastly, we need to mention that this experiment needs to be repeated in order for the results to be reliable. And another point is you want to be able to compare your results so you can make that final conclusion. So starting off with the independent variable, the thing that we are changing is the antibacterial solution. Now, the next part is the dependent variable. So the purpose of this investigation is to see which antibacterial solution is the most effective. So what we can do is we can have two concrete slabs. We can wash them with the antibacterial solution and then use a sterile cotton swab or a sterile transfer loop in order to transfer the bacteria that's on these two concrete slabs onto a Petri dish. And we would incubate it for some time to allow for that to grow. And then we can count the colonies that are growing on that Petri dish. So the dependent variable here would be the number of bacterial colonies on Petri dish. The next thing is your control variable. What are some things that you want to keep constant because that would affect the results of your experiment? So one thing is you want to make sure that the concentration of the antibacterial solutions are the same, right? It wouldn't be fair if one is more concentrated than the other. Another thing that you want to keep constant is the volume of the antibacterial solutions, right? It wouldn't be fair if you washed one concrete slab with more volume of antibacterial solution than the other. Okay, now for the method. So what we can do is we'll wash the two concrete slabs with the two different antibacterial solutions, and then afterwards we could leave it to dry, perhaps for about 10 minutes, and then we can transfer the bacteria that is on the concrete slab to the Petri dish using a sterile transfer loop or a sterile cotton bud. And the reason why it's important for it to be sterile is you don't want any bacteria on the transfer loop or on the cotton bud that would affect your investigation. So once you transfer that onto the Petri dish, you would then incubate the Petri dish for some time, maybe for about a day. And then afterwards, you would count the colonies that are on the Petri dish. It's always important to repeat our experiment to improve reliability. And then lastly, compare your results. So the one with if there's more colonies, actually, let me write this down the other way around. If you have less colonies, then that would mean that it's the most effective. Oops. Most effective. All right. I'm going to erase this. I'm going to write out the answer. All right. So first, wash two separate concrete slab with the two different antibacterial solutions. Make sure that the solutions have the same concentration and same volume. All right. After washing the concrete slab, leave it for 10 minutes, then use a sterile cotton swab to transfer the bacteria from the slab to a Petri dish. And then afterwards, incubate the Petri dish for 24 hours. And then count the number of bacterial colonies. Okay, and repeat the experiment to increase reliability. So technically this sentence here is not part of the marking scheme, but it's important to get into a good habit of writing that out. And then lastly, the antibacterial solution with the least number of bacterial colonies would be the most effective. All right, let's go over the marking scheme. Okay, so the first point for the marking scheme is equal volume of this solutions, same concentration, that's another mark. Wash the concrete slab with the solution. So I wrote that there. Leave it for a specified time. That's another mark. Use a sterile transfer loop or equivalent. So I wrote sterile cotton swab. And then another mark is transfer bacteria from slab to petri dish. Another mark would be incubating it for a specified time, so I wrote 24 hours. And the reason why is if we incubate it for too long, it might result in bacteria or pathogens that we might not want. Another mark is count the number of colonies. And another mark says repeat with second solution. So technically I mentioned it in the beginning when I said wash 2 separate concrete slabs. And lastly, compare the results. So I wrote, so for my last sentence, I basically said the one with the least number would be the most effective. And that's the comparing part of the marking scheme. All right. Let's move on to question 6. The diagram shows a white blood cell engulfing a bacteria and the stages that follows. So here we have the bacteria and you can see it is engulfing it. Part A1 says state the name of the process. The process is called phagocytosis. All right. Part 2, describe what is happening at stages 1, 2, and 3 shown in the diagram. So this part here would be stage 1. We can see a lysosome contains digestive enzymes. And what happens here is this lysosome would then fuse with this phagocytic vacuole. So we can write that down. The phagocytic vacuole fuses with the lysosome. Now let's see what's happening in stage 2. So in stage 2, we can see that bacteria is being broken down by the enzymes. So we can write the bacteria gets broken down by the enzymes. All right, and lastly, stage 3. We can see in stage 3 here, the remnants of that bacteria gets removed from the white blood cell. So let's write that down. The remnants of the bacteria is removed from the white blood cell. All right, part 3, suggest the function of structures labeled X. All right, so this is what they are referring to. So X is actually an antigen, and an antigen helps our body recognize the bacteria so that next time the bacteria reinfects us, we are able to identify it faster, and therefore the immune response would be faster. All right, let's write that down. Structure X is an antigen which is used by our immune system to recognize pathogens so that next time they reinfect us, the immune response would be faster. And even though this is not part of the marking scheme, but you can even add on to that, the reason why the immune response would be faster is because when we have an infection the first time around, we produce cells called memory cells. And memory cells are there to remember particular antigens, specific antigens, so that when the infection comes around the next time, we're able to produce a faster immune response. All right, so in the marking scheme, let's see, what does it say here? It says it's used to help identify or recognize pathogens. And the second mark is so that the response would be quicker. All right, let's go on to part B. A student observes a prepared slide of blood under a light microscope. The student draws a diagram of what could be seen. So that's their drawing. There's a really good drawing. Part 1, describe how the slide of human blood could be prepared. So I've included a diagram of a blood smear here. You can see that there's so many red blood cells. And then here we have some white blood cells. And the only reason why we're able to see the nucleus is because they've stained it with a stain. And so that helps make the nucleus more visible. So in order to get the human blood slide prepared, you need to prick the skin in order to obtain the blood. And then on that slide, so imagine you have a slide, imagine you have a little droplet there. You want to smear it so that the layer is thin. And then you would add a stain so you're able to see it, and then you can then look at it under a microscope. All right, so let's write that down. First, the blood is obtained by pricking the skin with a sterile needle. So the reason why it needs to be sterile is because if there's bacteria on the needle, then you're introducing bacteria into that person, which you don't want. Transfer a drop of blood onto the slide and spread, sorry, my handwriting. Spread it thin. Afterwards, add a stain onto the blood smear. All All right, let's look at the marks. Number 1, prick the skin. Number 2 is using a sterile needle. Number 3, we would spread the smear thin or spread the blood thin and then add a stain. All right, let's go on to the next question. Discuss the accuracy of the student's diagram. Okay, let's scroll back up to the student's diagram. Okay, let me bring out the smear photo again. So I've included a different diagram here, but this is what our blood would look like under a light microscope. And one of the things that we see here is in this diagram, our red blood cells that we see here are smaller than our white blood cells, whereas in this person's drawing, the white blood cells are smaller, right, which would be incorrect. Another thing we can point out here is that the red blood cells have a very different shape. It kind of looks like an elongated rectangle, whereas here in this diagram, you can see they are circles. So this diagram, the red blood cells appear circular. And lastly, your platelets, they don't really show up under light microscope because they would be too small for the light microscope to be able to detect them. All right, let's go and write that down. So first, the white blood cells in the drawing should not be smaller than the red blood cells. Secondly, the blood should be circular. Should not be any platelets as they are too small to be detected by the light microscope. Okay, part 3. Explain why the student would be advised to use a prepared slide rather than making one from their own blood. So to prepare a blood smear, you need to prick your fingers with a sterile needle, right? And one of the diseases that is known to spread because of the misuse of needles or sharing needles is AIDS, if you recall. So AIDS is a disease that is caused by a virus called HIV, which stands for human immunodeficiency virus. And that's why the student would be advised to use a prepared slide rather than using their own blood, because it would reduce the chances of getting these infections. All right, so you'd say the student is advised to use a prepared slide as it would reduce the chances of getting an infection such as AIDS. All right, let's move on to the last question. That's question 7. A method that shows whether a person's kidneys are working correctly is to measure the volume of filtrate produced by the kidney in 1 minute. This is known as the glomerular filtration rate. Let me show you what this is referring to. So if you recall, in our kidneys, we have multiple nephrons. And in the start of the nephron, you have this Bowman's capsule and the glomerulus, which is a bunch of capillaries that are found here. And what happens is that all these small substances like glucose, amino acids, ions, water, urea, all of that gets pushed out into the Bowman's capsule, and that's referred to as the filtrate. A person with healthy kidneys has a GFR of over 100 cm3 per minute. The GFR of a person whose kidneys were not working correctly was measured once a year for 9 years. Okay. We have the years, so that would be the x-axis, and your GFR in centimeter cube per minute, that would be the y-axis. Let's start off doing the x-axis first. So, barely have any space there. How many years do we have here? 2011, 12, 13, 14, 15, 16, 17, 18. 11, 12, 13, 14, 15, 16, 17, 18. I guess, let me just zoom out here. We'll have year over here at the bottom. What we can do is maybe have 2010 here, and then let it go up in 2. So 2, 4, 6, 8, 10. Yeah, I think that should be fine. So this would be 2012, 2014, 2016, 2018, and 2020. And then now let's look at the y-axis. The smallest number is 35. The largest number is 80. Maybe we can start from, say, perhaps we can have it start from the bottom. So we can write down GFR in cm cube per minute. And then we can have it go like this. So 0, 10, 20, 30, 40, 50, 60, 70, and 80. All right, so 2011, that's there, would be 80. 2012 would be 75. 2013, that's this one here, is 79. 2014 is also 79. 2015, that should be between those 2 years, 67. 2016 is 60. 2017 is 50. Then it goes down to 45. And then it goes even lower to 35. All right, and the next thing you want to do is connect these points together. All right, so let's connect this with a straight line. So make sure you use a ruler for this. Okay. So for this, we're just connecting the dots together. So there won't be any line of best fit here. We're just connecting the dots. All right, let's go to part 2. Explain why it is not possible to determine from the data when the person's kidneys stopped working correctly. Okay, so let's go back up. In the second paragraph, it says a person with healthy kidneys has a GFR of over 100 centimeter cube per minute, and this person starts off with 80, and that's the highest, and then it ends at 35 at 2019. So we don't know what year it is when it gets lower than 100 cm cube per minute. So let's write that down. the person's GFR in the beginning is 80 cm cube per minute, which is lower than 100 cm cube per minute. The data does not show at what year the patient's GFR drops below 100 centimeter cube per minute. All right, part 3. A person needs dialysis if the GFR falls to 15 centimeter cube per minute. Use the data to estimate when the person will probably need to start dialysis. So let's go back up to this graph here. All right, so when it hits below 15, that's when the person needs dialysis. So I'm just going to draw a line here. And then we can see that there is a gradual drop. It's not very constant because it's not a straight line, but you can see every year it drops for about like 10 cm cube per minute. You can see the drop here, right? You can also see the drop here as well. But again, it's not constant because in this 1 year it falls only 5 centimeter cube per minute. So if we predict what year this person might need dialysis, let's assuming it's 10 centimeter cube per 10 centimeter cube per year. So that means it's going to drop like this in 1 year and this in another year. So that will be 2021. All right, let's write that down here. So from what year is it? From maybe around 2015 to 2019? 2015 to 2019, the GFR decreases by 5 to 10 centimeter. Let me just write the centimeter cube together. Centimeter cube per minute. Therefore, the person might need to start dialysis at 2021. And we'll say to 2022. All right, discuss the accuracy of this estimate. All right, let's scroll back up. So I'm just going to zoom out a little bit here so we can see. All right, so we can see that over the years, starting from 2014, the rate at which it decreases changes, so it fluctuates. So you can see here from 2016 to 2017, there was a large difference, but from 2017 to 2028, there was a smaller decrease. That's one thing you can say is it's not very accurate. Another thing you can also say is in the beginning, this person's GFR decreased and then increased again. So it is a possibility that this person could have their GFR increase, right? So again, it's not very accurate. So let's write that down. All right, so we can say the data is not accurate as the rate at which the GFR decreases starting from 2014 fluctuates. So that's one thing we can say. And another thing is during 2011, so 2012 it decreased, and then 2013 it improved. This person might also have their GFR improved like it did between 2012 to 2013. Where the GFR increased. Right. Lastly, let's go over the marks first. So the first mark is data isn't accurate because of the fluctuations. That's one mark. Another mark is so earlier data shows that there was improvement, right? And I quoted the year. And then the third mark would be that might happen again. Right, and I sort of implied that in the last sentence. Okay, let's go on to part B. During the early stages, when a person's kidneys are not working correctly, they are advised to control the amount of protein in their diet. Explain why protein in their diet should be controlled. So our kidneys are responsible for removing urea, which is a waste product that is produced due to the excess of proteins. So if we consume too much proteins, what happens is that proteins gets converted to urea, and then that would be removed by the kidney. But if the kidney aren't working, then the person will not be able to remove urea properly from their body, and that would be toxic for them if it builds up inside of their body. Okay, so the kidneys are responsible for excreting urea from the blood. Urea is produced during the breakdown of proteins, and that process is called deamination. If urea is not removed, the build up urea would be toxic to the body. Therefore, if a person's kidneys do not work, they need to reduce their protein intake so that there is less urea. All right, let's go over the marks. One mark is proteins are broken down to urea. That's one mark. Another mark is that it is toxic when built up and it needs to be excreted from the body. And then a person would need to consume less protein intake so they would have less urea. All right, that's it.